apparently going to talk about. There we go. Just remember that the hybrid possibilities of outcomes from hybridization, right? One of them is we get low fitness hybrids, which creates selection for pre-zygotic isolations, which we call secondary reinforcement. Second possibility is hybrids are super fit in some place and become isolated from their parental stocks. So we get hybrid speciation, a new lineage that arises out of that hybridization event. And the third I'm not going to spend much time on, really any time, is just the idea that we may maintain these things called stable hybrid zones. And that's just a place where the parental stocks come together, they hybridize. The hybrids are not so unfit that they're completely selected against, but they're also not so fit that they spread in space. And they're not isolated from their parental stock. So you get back crosses, you get hybrid crosses, but they never really move in space. And we see this in all kinds of places. This is a graphic from a European toad, a fire-bellied toad. The yellow-bellied species and the red-bellied species are reproductively isolated, but there's a line through Eastern Europe where hybrids are formed. And you know, that's not, you can see that's not an even line. Right? What is it that makes it go where it goes? We don't really have a good handle on it. Sometimes it's an altitudinal zone, sometimes it's a climatic zone. But if you were to look at an allele frequency, let's say it's an allele from the fire-bellied toad, it's fixed in the fire-bellied toad as you move through space, so this is distance, and you get close to the hybrid zone, you're going to have this very short distance where you've got intermediate allele frequencies. And then when you get to the other side of that zone, you're going to have none of that allele because you're in the yellow-bellied toad range. So what happens is these hybrid zones become a place where alleles can move from species 1 to species 2, introgression. I think we talked about it. We'll talk about the process again in another context. But just remember that this is a third possible outcome. And I'm not going to ask a whole lot more than that. Just remember that one of the possible outcomes of hybridization is a stable hybrid zone. All right, we didn't talk about ecological speciation, although it's turning out to be super important. The thing that makes ecological speciation different than the other processes that we talk about is one key piece. Selection for some kind of a phenotype is the thing that creates reproductive isolation. So not a physical barrier, not some sudden event like a polyploidy arising through a meiotic disjunction, but selection for some trait that causes reproductive isolation in and of itself. And this is, I've mentioned this word I hate, or phrase I hate, called magic traits, but I think it might help you get the idea. The trait's thought to be magic because selection favors the thing and the trait itself creates the reproductive isolation. So what could possibly do that? A really good example comes from mimetic butterflies. So this is a species complex found in Ecuador. Over on your right, you've got Sappho and Alukia. These are butterflies that feed on a host plant that gives them a compound that's really nasty. So birds don't like to eat them. And birds recognize white-winged butterflies and yellow-winged butterflies as nasty things. But all three species, so Sappho, Alukia, and Cydno, all live in the same place. And Cydno is splitting into two species. There are two color morphs. One that looks like the yellow-winged butterfly and one that looks like the white-winged butterfly. So selection favors the color pattern morphs because they look like these nasty tasting butterflies. They're actually also nasty tasting. So how do they become isolated? Why don't they interbreed and just blend the yellow and the white back together, or one of those morphs takes off? It turns out that this is a magic trait for butterflies because butterflies recognize their possible mates through color pattern. And so if you ask a yellow Cydno here, who would you like to mate with? She would prefer to mate with another yellow butterfly. A white Cydno prefers to mate with a white butterfly. And this is the distribution of mate choice that comes out if you ask those butterflies who they'd like to mate with. So it's magic in the sense that it's selected, it's favored from an ecological point of view, but it also creates this instant reproductive isolation that can split the species. So we think this is an example of speciation on its way in Ecuadorian butterflies. So there are lots of really good examples now of ecological speciation. Some of the best ones are in plants, but this one I think is clearer because we can look at the color and we can see why it might work. All right, so if we play up speciation, like we've talked about like 1 becoming 2, how does that turn into biodiversity? I started this whole section with pictures of cichlids, because I think they're really cool, but because they're also a great example. So here's Africa, here's the Rift Valley. If you went right down here, this is what's called the Rift Valley. These big dots refer to these lakes. Lake Victoria, Tanganyika, Malawi. Each one of those lakes has a species flock of cichlids. Depending on the paper and the lake, that's between 600,000 and maybe 4 million years old. And each of those lakes was invaded once by a riverine cichlid, something that's not too different from tilapia, which is banned from my house because it tastes nasty, but it's the ancestral cichlid type, got into these lakes and then exploded. And so these are 3 examples of adaptive radiations, which we think of as explosions of diversity in a very short geologic time. So I'll show you what that looks like in a phylogeny, but the important idea is one branch that if you looked in a cross-section of time becomes many branches all of a sudden. And then they stay pretty much stable after that. So in the cichlids, what you've had is this repeated radiation of the same ecomorphs, or the same types in each of the lakes. And I just called out a couple of examples that I think are really cool. They're all labeled with the same letter. So the scale biters, the ones that swim up next to other fish and take a chunk out of their scales, that's B in each of these groups. So here, here, and here. They all have mouths that kind of go to one side. There are things that crush snails. They're pelagic fish eaters, piscivores. They look more like something we call a pipe-shaped cichlid, right? That's the H. Repeated invasion of the same set of niches that are open, one species becoming many, many species. That's an adaptive radiation. So if we look at it in a more abstract sense, what we're trying to identify are places where that diversification coincides with that open niche or open space. It could be an island, it could be a lake that's never been invaded, and has all these open possible niches for new species. It leaves a really obvious pattern on a phylogeny, and it's the pattern that I've circled here in the middle, or boxed in the middle. So here, we imagine all speciation events are bifurcating, so 1 to 2. But if you drew the line, or drew a box around this series of events back in time, that's a pretty short time where we went from 1 to many species. And then there was a long period of pretty much stasis, and then another event here where all the branches are kind of in the same place. And on the bottom, if you plot the number of lineages on the Y and time, you see a rapid increase in lineages, and then it kind of stabilizes, and then another rapid increase, and it stabilizes again. So that's the hallmark of an adaptive radiation. Contrast that to the thing on the left, which is a randomized series of branches. Even though it looks in this plot like you've had a quick burst in the beginning, this is really a random pattern. There's no, if you look up here, there are branches that come off at any given time. On the far right, you could actually call that another kind of adaptive radiation. It's just much more recent. You had a long period of stasis, no speciation, and something happens that opens up all these niches. So the best example, or one of the best worked examples of this, are anole lizards from the Caribbean islands and Greater Antilles. So what seems to have happened is some ancestor, probably from a South American mainland, got onto Caribbean islands. So we're talking Jamaica, Puerto Rico, Hispaniola, Cuba. And on each of these islands, they invaded the same set of niches. So we've got a trunk ground anole like this that sits on a branch headed down, great big legs. We've got things that live up in the canopy, crown giants like this that are bright green, slightly shorter legs. Things that are cryptic little twig crawlers, so they got really short legs and long limbs, and they move like this, like a chameleon. You guys are hard. It's not that early. Dance like a chameleon. And grass bush anoles, right? So these are all different islands. So even though they look the same, this is a Cuban anole, this is a Puerto Rican anole, this is a Jamaican crown giant, a Puerto Rican crown giant, right? So what happened, like I said, is some ancestor came from the mainland, which is off box here, and invaded different islands. And if you look at the tree, it probably started on Hispaniola. But anyway, these silhouettes are the islands. So here's Cuba in red, Hispaniola in blue. Hispaniola's Haiti and the Dominican Republic. Puerto Rico here in kind of dark orange, and Jamaica in purple. So on the bigger islands, the pattern of adaptive radiation isn't quite as obvious. So here we've got the number of species again on the left, time on the right. On these two giant islands, it looks like there's an initial burst and then it's pretty stable. Right? And that's because they're giant islands. They diversify and then there's speciation within the island, and they diversify again. On the smaller islands, or what we think of them as big islands, but they're smaller on the scale, you had one initial event where one species became about 5, and that was it. Right? There's almost no more speciation on these in Puerto Rico and Jamaica. A very small flock. So that's the classic adaptive radiation. Quick burst, long period of stasis. If you look at those on a tree, it's a bit messy, so I want to call your attention to just a couple of spots. These are most of the greater Antillean islands and anole species. So look down here in yellow at the Jamaican group. So here's Jamaica in yellow, Jamaica in yellow. You don't see any more yellow. All the species of anoles on Jamaica came from one ancestor. They speciated really quickly in time, and they didn't go anywhere else. Right? Look up here in dark brown, right? This is Puerto Rico. It's a little more split in time, but they all line up with the same common ancestor, and most of the speciation happened very early. Go to the big islands, like the purple, Cuba. You had an origin here where you had one species that got there and diversified, it kind of spread out over time. Here's another lineage that got there later on, diversified. So if you pick it apart and you look really carefully at the phylogeny, you see multiple adaptive radiations within the big islands. But the pattern's easiest if you look at these smaller clades, like the yellow and the dark brown. Okay? So that's adaptive radiation. We're going to come back to it when we talk about coevolution for a particular kind of radiation. But again, one species becomes many in a short period of time, filling up new ecological niches. Good? All right, we're going to move to coevolution. So coevolution, there's no real chapter in your book. I'm going to post up a review paper that's, it's maybe 8 pages long, and it's kind of meant to be a primer on coevolution. And I'll see if I can get a copy of another textbook's chapter that can't really be posted, but maybe it will. So there's no reading to go along with it, so it's important that you go with the notes. So this is one of the things that I like and I work on. I want to start you with, let's go back to review. I'm going to start you with the basics. Why is it coevolution? Why is it different? Talk about some dynamics. So these are 2 of the possible outcomes of coevolution. And then we'll talk about some specifics and manifestations in different systems. So coevolution is different than all of the other evolution that we talk about because it's a biotic force of selection that changes. So that means we can evolve in response to climate. We can watch a species evolve into increasing temperature. Right? And that's an abiotic factor. The species evolving doesn't change the climate for the most part, unless the species is human. But if you talk about hosts and parasites, species one evolving changes species two, and that creates this feedback loop. And that's what makes it different and kind of fascinating. So imagine this as your general picture. We've got species A and species B. I've drawn them as enemies, predators and prey. The ability of the predator to chase the prey creates selection on the defensive ability of the prey, right? If the classic cartoon is the fox chasing the hare. As foxes get faster, the hare's a rabbit. The bunny has to get fast to get away from the fox, all right? As the fox gets faster, the ability of the bunny to run increases, so it gets faster. But as the bunny gets fast, the fox now experiences selection to get faster, right? So we've got this reciprocal feedback loop, and we get the evolution of faster foxes. And that drives faster bunnies, and on and on and on. Right? So what is it about coevolution that's different? It's this word reciprocal. Right? And there are two things that have to happen for reciprocal evolution. You have to have selection that is linked between the two species. The trait that's being selected by species A has to create selection back on species A. Right? So speed and speed. I'll show you an example of resistance to a poison and the poison. So as you get more poison, that selects for higher resistance. The second piece comes out of the famous R equals H squared S formula. Response is heritability times selection. You have to have genetic complementarity. So it doesn't do any good if there's no genetic variation for bunny speed. You're not going to get coevolution. So you have to have genetic variation in both traits on either side of that interface. So that's the general picture of coevolution. Here's a case of it in a little more detail. So it's a beautiful example because it's physical features that you can really kind of get your head around. This is camellia flowers, so these are camellias like grow outside the greenhouse behind Gilmer Hall or in your mom's garden, your dad's garden. Camellias have a fruit with a seed in the middle and a thing called a pericarp. That fat part on the outside is basically a protectant. Weevils are a beetle that are pretty crazy looking. Right? So here's the body of the beetle. This is its mouth part. It's called a rostrum. So it's like an elephant's trunk, except it's a long snout, and at the end of its snout is its mouth parts. So there's little teeth out here. And females chew a little hole into the pericarp of the camellia. So if you look closely at the picture, this is the hole. This is a split open camellia fruit. This is a hole that a female has chewed down, and then she turns around and lays an egg in the hole, and the larvae grows up in the seed. So what's the selection? If a female weevil's snout is not long enough, she can't reach the inside of the fruit. So her egg is got a dead end. If the pericarp of the camellia is not thick enough, it can't prevent this parasitoid from digging all the way through. Right? So you've got basically matching of the thickness of the pericarp and the length of the rostrum. Right? And this is a beautiful work done by a couple of Japanese scientists, Tojo and Sota, where they did all the functional relationships between the rostrum length and the pericarp. And what you find is, sure enough, longer snouts produce higher fitness, so red's higher fitness, when the pericarps are shortest. So the best thing for a beetle is if you get a thin wall and a long snout. The best thing for the seed is to have a thick wall and a short-snouted weevil. So what do you think happens? Over all these islands off the southern tip of Japan, you find matching levels of pericarp thickness and rostrum length. Right? So we think of that as an arms race, and I'll talk about arms races in a second. Okay, so there are actually quite a few different kinds of dynamics that coevolution can end up in. I'm going to focus on two because they're kind of the big ones and obvious ones. The first one we talk about as an arms race, and it's kind of an old Cold War metaphor. But it's the idea that enemy one has escalates its defense, so enemy two has to escalate along with it, and as enemy two escalates, enemy one escalates, right? So this is the old Cold War. You get a missile, I get a missile. You get a bigger missile, I get a bigger missile, right? So what does that look like in evolutionary time? If you take species one, I call them prey here, species two, predators on the Y, and we have a starting point, what's going to happen? They're both going to get pushed up. Right? So you get an arrow that moves up. It moves up. Just have to have a little more English. That's one generation. What happens the next generation? Moves up again, right? And then another generation, increase, increase. This is the reciprocity, and it just keeps going and going. When does it stop? It may never stop. It may stop because one of the species experiences such an enormous cost to producing this trait. So let's say it's a poison in the skin of a newt, the story I'll talk about. Maybe there's so much poison in the skin of a newt at some point that it becomes toxic to itself. Or maybe it's maybe there's some other cost to the predator that there's no ability to get to the end of the functional ability to resist the effect of that poison. So at some point, maybe a cost ensues, or one species is driven extinct and the other's left. Right? But there's no change in that trajectory. It's just escalate, counter-escalate, counter-escalate, counter-escalate. We think this is most important for quantitative traits where you can imagine kind of a more is better scenario. So with the camellia and the weevil, a longer snout's better, a bigger pericarp's better. Right? Having a different shaped pericarp isn't going to change the dynamic, but having a longer pericarp does. Right. So contrast that with this other dynamic, and I know you talked about it a little bit, called the red queen, and this is from Alice in the Looking Glass. The red queen has to run faster and faster to stay in one place. This is the idea that we essentially have negative frequency dependent selection on something that's involved in this interaction. I think this is important for things like host-parasite interactions. So these are cases where maybe you've got a qualitative ability to resist a predator or a parasite. So the real dynamic or the real functional interaction that makes sense here and is shown to happen with a lot of host-parasite interactions is there may be some protein that has to be recognized in the parasite that the host immune system recognizes and attacks. And as the host system learns to recognize that parasite, new parasite types have an advantage, right? So it's like having a new code. The old code gets broken, you need a new code. And if the new code becomes common, the host better break that code, and then a new code becomes common. So what happens is an oscillating dynamic. So if you look here, this is what this will look like. So here I've got the parasite. As the parasite gets common, the host matching type starts to track it. There's a lag. That's important. They're not matched in common. One of them becomes common and then the other starts to kind of chase it. So if we say what's the allele frequency, the parasite frequency goes up, the host goes up, and it's maybe a generation or two behind. And then as that parasite code becomes the most common, it gets cracked. And now it's not fit anymore. And if you watch this, what you're going to see through time is this oscillation, right? One becomes common, becomes rare. Now it becomes common again. As the code gets cracked, it drops in frequency. But now it's rare, so the hosts don't recognize it. So this is what we think of as this oscillating arms race that we think is probably very important in disease and host interactions because of that qualitative interaction. Match this protein with this protein. Having more of it doesn't help. So it doesn't really make sense to fit an arms race. Having a different one is what counts. And that's one of the key differences between when we predict an arms race happens and when we predict a red queen dynamic happens. Okay? So a couple examples. Oh, sorry. One more general point, which is part of the example. We often think about coevolution as species A and species B. And the reality of ecology is that what goes on locally is what drives evolution. And this is a relatively new perspective on coevolution. So I'll show you maps, but imagine a distribution of two species that interact, predators and prey. Well, in some places, maybe the coevolution is really tight. Right? There aren't any other prey to eat, so the predator's really locked onto that one. But you go 200 miles away, and there's some other prey to eat, so now the predator doesn't rely so much on that prey item, and maybe you've got something like this, where there's not really intense selection. And maybe there's another place over here where really there's no selection at all. They don't even interact. You know, one of them lives in this pond and the other one lives up on the hill. So there could be a lot of variation in a small spatial range of whether or not you get intense coevolution or not. You know, this could happen with hosts and parasites. It can happen with pollinators and plants. You get a different pollinator in the system. Now the coevolution between bumblebees and the flower is really weak when we introduce honeybees. Okay? So the point really is that we get a range of variation. So some places there's no coevolution. Some places there's lots of coevolution, really strong. And what do we know from gene flow? Well, if we get the evolution of, say, resistance to a poison in a hot spot, what's going to happen with those genes if there's gene flow? Some of those genes are going to move out into a cold spot. And the outcome is that we're not going to get perfect matches because those genes are going to move from place to place. And so one of the predictions of this mosaic is that we get matches and mismatches, and you have to look at the ecology in each individual spot. And let's be perfectly clear, because nobody ever asks questions. Okay, so here's a story, an example. This is one that I've worked on with my lab for a while. So if we think about predators and prey, this is kind of the classic cartoon coyote and roadrunner example. And there's a reason to think that it's not a good example of coevolution, and that's this idea that selection could be asymmetrical. So think about the interaction between the coyote and roadrunner. You may not even know that cartoon anymore. Is that even, you guys know Coyote and Roadrunner?
Yes.
Yeah, oh, because you got YouTube, right? That's how you watch it. So this is not Coyote and Roadrunner. This is a vine snake like Tophus and a lizard anole. Who has more to lose in this interaction? Individual. The snake or the lizard? The lizard, right? Because if the snake loses, it can go hunt again and it can find a meal another day. The lizard loses, what's its fitness from that point on? 0. Right? So that's an asymmetry. So there's strong selection against the prey being caught, not as strong selection for predators. So this is the argument anyway, that there's an asymmetry, and it's referred to as the life dinner principle. The predator loses its dinner, but the prey can lose its life. So if you amortize that over all the individual interactions, the population of prey should evolve faster than the population of predators. Maybe. But the prediction is if we look across species or across populations, if we see one species getting out of the arms race, it's going to be the prey, not the predator. Okay. So we looked at this interaction, garter snakes and newts. Garter snakes are the pinnacle of vertebrate evolution. This is a western garter snake eating a newt, a rough-skinned newt from Oregon. Rough-skinned newts are they're newts. You know, they don't do anything. If they're threatened, they do this behavior where they curl up and show you their red belly, orange belly. And they're advertising this thing called TTX, tetrodotoxin, that's a channel blocker. So if you remember anything about your basic neurobiology or vertebrate physiology, sodium channels in the membranes of nerves and muscles are critical for they allow sodium into the nerves and muscle, and that is a key piece of propagating action potentials. So without sodium channels functioning, you're dead. Right? So this is a lethal toxin. These guys have enough to kill maybe 10 to 20 humans by weight. So every year or two, there's a little news article in Oregon or Washington where somebody, usually male, usually involves alcohol, has decided to eat a newt on a dare. Not a good thing. So don't eat newts. This is the key piece of the lecture today. Don't eat a newt. You will die. What we know about snakes is that in a lot of places they've evolved resistance to that toxin. And I'm not going to go into much detail, but this is the molecule, the sodium channel, and there are substitutions here, single amino acid substitutions that block the binding of that toxin. Okay, so snakes evolve resistance. If you look at that over a landscape, you can draw pretty maps, and you can see the geographic mosaic in action. So here's the strength of toxin in newts, so how much of that TTX do they have? Hot colors means more. Here's the resistance in snakes. Hot colors means more resistant. You can see it lines up pretty well. You got a matching up here in Oregon, a matching down here in California, in the Sierra and the Bay Area. Right? So more toxin in the prey, more resistance in the predators. That's a pretty good picture for arms races. But it masks some of the nuance of what goes on. Right? So if you dig in a little more deeply and you ask, well, how much toxin does it take to kill a snake or knock down a snake? And how resistant does a snake have to be to eat a newt? You can draw a graph like this where this orange area is sort of variable outcomes. You know, some of the newts will kill the snakes, some of the snakes can eat a newt. We can't really predict what the answer is. But below that line, snakes are so resistant that they're not affected. And above that line, newts are so poisonous that they kill every snake. So, you know, newts win at the top, snakes win at the bottom. And each of these points is a place. And you can see that about half the places they're matched, and about half the places they're not matched. So this fits that geographic mosaic idea that maybe gene flow has moved genes that work well over here over to a place where they don't work so well. And if you redraw the map, let me back up. Back up there. Hold this picture in your mind. Red here in Oregon, orange down here in the Bay Area. That's the amount of toxin or the amount of resistance. But if you plot that on a functional level and say, is that enough to knock out a snake or enough to knock out a newt? The answer is no, right? So there are still hot spots, but they're not in the same places. So we find places where they're really mismatched, the cold colors, and where they're tightly matched, the orange colors. And it says that, yeah, there are hot spots and cold spots of coevolution, and it's not necessarily where the values are the strongest. So look, especially right here. This is a place where it turns out to be the Bay Area, California. Snakes are super resistant and newts are super toxic, but the snakes are so resistant that you literally can't afford enough toxin to knock them out. And the newts don't have enough toxin to kill a snake. Right? So snakes have won. So the life dinner principle didn't work. That's part one. We had a nice prediction that didn't work out very well. Maybe it's because instead of just costing the snakes a meal, they actually can kill the snakes. So that erases that asymmetry. Right? The other question is, why do we end up with that jump? Why do the snakes win and not the prey? Right? So this is the same plot with a cartoon to show you what we think happens. So we start out with an ancestral condition, right? This is the ancestral newt before they got into the arms race and the ancestral snake before they got into the arms race. Something happens that drives up the toxicity of newts, and it gets into that orange band. Right? And now we've got that arms race dynamic of up and up and up and up. Okay? Right? Arms race ensues, there they go. Newts get more poisonous, snakes get more resistant. Up we go, and then something else happens, and boom, they pop out into this snake win zone. So what is it that creates this green jump from arms race coevolution to predators win? And the punchline seems to be that we know from some experiments that single amino acid changes change the resistance tenfold. So it's not the gradual evolution that you might think of as a little bit more and a little bit more. That's this part of the interaction. A little bit more resistance, a little more toxin, a little more resistance, a little more toxin. And then you get one switch of an amino acid, a single amino acid in that pore, and it goes from 100x resistance to 1,000-fold resistance in one, it's actually one base pair change and one amino acid substitution. So we think of that as a saltational evolutionary step, a jump. So it's just the plausible explanation right now for why that happens. But it's a good example of the geographic mosaic and the arms race in action in predators and prey. All right, so there's a question. If you follow a geographic mosaic, what's the general prediction from the geographic mosaic? You can tell how far behind I am because we're getting one of these a lecture instead of two or three. Let me turn on. You are the quietest group of 300 people I've ever heard or not heard. All right, you guys must like newts and snakes, because this one seems like it's working out pretty well. Pretty well. So think about the geographic mosaic. Take it away from newts and snakes. Think about a general prediction of coevolutionary strength that changes in space. Is there at least thinking more carefully about this one? All right, good. You guys are pretty well on it, right? So this is a classic example of a multiple choice question that gives you plausible answers, right? They're not false statements, but they're not relevant answers, really. So the geographic mosaic doesn't tell you anything about whether prey or predator should win. That's really about the life dinner principle, so it's a different thing. C is plausible as an outcome, but D is really what's predicted. Right? That the strength of reciprocal selection varies. Sorry, C is the answer. D is part of the geographic mosaic, but C is what is an outcome. That we could get movement of genes back and forth through gene flow. Does that make sense? You guys, yeah? Cold spots could turn into hot spots, but it would require a change in the ecology. Sorry? Yeah, right. So what could happen is cold spots could move genes of low resistance into an area of high resistance, and that would change the interaction. D, the strength of reciprocal selection varies depending on the local ecology. That's part of the mosaic, but it isn't a prediction of the mosaic. It's why the mosaic happens. Right? Questions are good. Okay? All right. Okay, so red queen, the other major dynamic, I know you've heard this example before, but it's really a classic. So this is a parasite of aquatic snails. It's been worked on in New Zealand by Kurt Lively and Jukka Jokela, great name, Finnish guy. For years and years. And the basic biology of this trematode parasite is that it reproduces in ducks, and then ducks poop out the eggs, and snails graze on algae, and they eat some eggs and some duck poop. And then the trematode grows up inside of a snail, and it's a castrating parasite. So it literally eats away the entire gonads and internal organs of the snail. That's a bad thing. Your fitness is pretty much zero. And then forms cysts that go out into the water, and the ducks ingest that when they're eating algae. And so it's a classic parasite that has two hosts. This is Potamopyrgus, the snail. These shells just tell you they don't look any different, but there are sexuals, males and females, that reproduce. They're hermaphrodites that reproduce sexually. And then they spin off these asexual lineages that are triploid, and they self. So they don't recombine, they're genetic clones. And so every lake has a combination of sexuals and a few clones. So you can think of the clones as different genes, right? So because they're genetically identical. So each clone in a lake is a genotype. And what the question is, basically, does the most common clone, the most common gene, have lower fitness than the rare genes? So if we have a red queen dynamic going on, what would we expect? We'd expect the rare gene to be at a disadvantage, at an advantage, and the most common gene to be at a disadvantage. And if we look across lakes, moreover, you would expect that the parasites that have evolved to the common clone over here are not very good at infecting the common clone over here. So there are two predictions. The rare genotype should have the highest fitness for a host. And second, you should have a home advantage if you're a parasite. You should be really good at attacking the most common local clone, but you haven't adapted to the most common away clone. So Kurt and his lab have looked at this for years and years and done a variety of experiments. The simplest punchlines are these two. Within a single lake, if you take the most common clone and compare it to a rare clone that's barely present, you find that the infection rate is much higher on all the common clones than it is on a rare clone. That's prediction one. The one that to me, really nails it down is that you should see that geographic variation. And this is another example of a geographic mosaic. Sure enough, if you do this home and away test, each of the colors is a different lake. These are names of lakes. So the P lake, Paringa, the P host does the best, but it doesn't do so well in the other lakes. The Wahapo lake, Wahapo? It's New Zealand, I don't know how you say that. The W lake infects the W host, but doesn't do so well in the others. So you don't have, this is important, you don't have one parasite that's best. The Paringa lake is, the Paringa parasite's not the best genotype. It's only the best in that lake. You put it against the host from another lake, it doesn't do so well. So that's that matching that is predicted from the oscillation of the red queen. They've done a number of other things that are multi-generation experiments, but this is really the pattern you would expect if you had red queen dynamic. Okay, so just to review arms race and red queen. The arms race is reciprocal directional selection. It's pushing up and up and up, right? Never changes, doesn't push down, doesn't select for a different flavor. It's more is better, more is better. The red queen is negative frequency dependent selection, right? So imagine a question that could arise out of that slide. What's the different form of selection that drives those two dynamics? It's negative frequency dependent because the rare type has an advantage and it increases. When it becomes common, it's disadvantageous and it's driven back down. When it becomes rare, it's get pushed back up in frequency. So that is the classic negative frequency dependence. When do we expect to see those two things? Top one, arms races, quantitative relationships. More is better. Red queens, qualitative or matching dynamics. Things like host parasites where you've got a lock and key or a code and a code breaker. So when you hear people talk about arms races between antibiotic resistant bacteria and antibiotics, it's really more likely that it's a red queen and not an arms race. And that stuff matters. Okay. A couple more, maybe I'll just do this example. So the examples I've talked about so far are enemies, predator prey, host parasite. We can also, everybody gets along, right? These are mutualisms where everybody gets an advantage, but we can see similar dynamics. So the most obvious one to most people is plant-pollinator interactions. And this one's beautiful. This is a South American hoverfly. This is the tongue of the hoverfly. Check this thing out. Right? You think the rostrum of the weevil is cool. Look at that. This is like 5 body lengths. Imagine if your tongue was 5 body lengths long. Curl that thing out. This is a flower. I think it's a geranium. Maybe it's an iris. Anyway, it's a flower. It has a really long nectar tube. And what's interesting about, you got the match, that's the obvious thing. What's interesting is you've actually got directional selection on floral tube length. So the flowers that are longest have the most pollen transfer efficiency. Probably because the fly has to spend more time in there digging around to get to the nectar, so that increases pollen transfer. So there is a directional selection advantage to the flower to have a longer tube. Okay? Similarly, for the fly, this is the tongue, the proboscis. The longer the tongue, the more nectar it gets. So it's getting more calories if it has a longer tongue. So this is directional selection on both, independently. But there's also a matching component, right? So the longer tongue is good for the fly, the longer tube is good for the flower. So if we dissect the mutualism, we have both directional selection components and frequency dependence. But mutualists are different than enemies. So now it's not good to be rare. So it's frequency dependent, but it's not negative, it's positive. Does that make sense? So are you the best, are you gonna have the highest fitness as a flower if you have an anomalous floral tube length? No, because you need to match the pollinator population that's out there. So you've got to have the most common pollen tube type. So that means it's positive frequency dependence. The most common one fixes really quickly, and then you've got this nice matching. So these are just diagrams of what's going on with the fly and the tubes, right? There's nectar at the bottom. But focus on this, positive frequency dependence, the most common morph or type of both the plant and the pollinator gives the highest fitness. So you end up with what looks like, it would look like the same dynamic as an arms race, but it's not the negative fitness consequence, it's the positive fitness consequence. So same sort of co-evolutionary dynamic. And I think, yeah, I'll stop with that and I'll pick up mimicry on the next one.